Alright, so today we're going to talk about meiosis, which is similar to mitosis but has some very important differences. Okay, so remember with mitosis, the purpose of it was to produce cells with the exact same information, make exact copies. And the reason that you wanted to do that, so they were exact copies, it had the same amount of DNA, same number of chromosomes, exact same information, clones, all exactly the same. Very important. Okay, why do cells do this? Well, remember that single-celled eukaryotes, this is the only way that they can reproduce. Okay, so there's some examples. And then simple multicellular or eukaryotes are also going to use mitosis as a way of reproducing. So this would be budding right here, where this is a hydra, which is a relative of a jellyfish. Okay, and it literally just grows its offspring out its side. It comes growing out of the side until it's big enough, and then it just goes and lives on its own. And you had to put two. Okay, so they're making clones. Remember, asexual reproduction has some very important advantages and some disadvantages. Advantages: fast, easy, make lots and lots of babies. Okay, that's good. Disadvantages: everybody's exactly the same. So everybody has the exact same chance of survival, the exact same chance of dying. And so it's very hard. If the environment changes, there's nobody among you that's different that might survive better than someone else. Now the rest of us, can we reproduce by mitosis? Well, remember, with sexual reproduction, you get information from mom and information from dad, egg and sperm. They join together, and they make baby. Right, if we made egg and sperm by mitosis, what would happen? Well, remember that human cells have 46 chromosomes in them. So you'd get a human egg cell with 46 chromosomes, a human sperm cell with 46 chromosomes, and you'd put them together and you'd get a 92 chromosome baby. Yeah, that doesn't work. Wouldn't survive, can't ever happen, don't ask me ever about it. It cannot happen, does not ever happen. Okay, so this does not work, does not happen. Here's some pictures of human chromosomes. This is called a human karyotype. It's literally a picture of the chromosomes from a human. And you'll see it's a female. How do we know it's female? Because the last pair are both X chromosomes. They're the same size, both X's. 2 X's make you a girl. Okay, here's the male karyotype, and we know that because of the X and the Y. Okay, that's really the only major genetic difference between males and females is, do you have 2 X's or do you have an X and a Y? X and Y male, 2 X's female. Alright, remember we talked about this when I first mentioned mitosis. We talked about homologous chromosomes. These are chromosomes that carry matching genes. You got one of them from your mom, one of them from your dad. Okay, they can show the same inherited characteristics, the same information, homologous. Okay, and when we're drawing chromosomes, we usually represent them as being the same size, but not necessarily the same color. Okay, so you can see that in this cell, there's 2 long chromosomes and 2 short chromosomes. So the 2 long ones are homologous to each other. They carry the same types of genes. And the 2 short ones are homologous to each other. They carry the same types of genes. Why are 2 of them red and 2 of them blue? Well, because this cell got the 2 red ones from its mother cell and got the 2 blue ones from its father. Okay? We refer to cells that have 2 sets of chromosomes as being diploid. Okay, so there's your single-stranded homologous chromosomes. They'll get replicated before meiosis occurs, and you'll end up with double-stranded homologous chromosomes. But again, carry the same information. All right, so in order to make sperm and eggs, somehow you've got to get cells to be half the number that they started out as. So in humans, 46 has to go down to 23. And then when you add 23 from mom and 23 from dad back together, you get 46 baby. Yay, that's good. Okay. So how does this happen? So let's look a little bit. Meiosis causes alternating stages in your life. Everybody started out as a diploid zygote. When egg and sperm got together, you were diploid. You grew through mitosis into being either a diploid male or a diploid female. In your sex organs, and only in your sex organs, meiosis has occurred to make haploid gametes. Okay? So this is the diploid portion of your life, and then this is the only portion of you that is ever haploid, are your sex cells, either your eggs or your sperm, depending on who they are. And then one day, far in the future, when you graduate from college, have a good job and a stable, loving relationship, one of yours may combine with somebody else's and make baby, but not for a long darn time, people. Okay, So you go from diploid to haploid and then back, combine with somebody else to make diploid again. All right, so diploid we call 2N because there are 2 sets of chromosomes, and then haploid is N, the 1 set of chromosomes. In humans, the diploid number is 46 and the haploid number is 23. In chimpanzees, it's 48 and 24. And everybody's a little bit different. Those are the 2 I remember off the top of my head. Meiosis is going to reduce the number of chromosomes in half and make gametes or sex cells. Okay, then fertilization brings them together and restores the chromosome number back to the diploid. Okay, haploid, diploid. All right, so gametes are haploid. Through fertilization, you make diploid copies, and then through meiosis, you make haploid ones. Okay, notice that this cell had, so this one was information from mom, information from dad to make baby. Then when this baby went around and started making its gametes, it made gametes that weren't just like the ones it had been made from, they were combinations. So we pass on different combinations of genes to different potential offspring. And that's good because it makes everybody a little bit different. Variation is a very good thing. All right, so remember that meiosis is specific special cell division for making gametes. Takes your cells from diploid to haploid. Okay, meiosis evolved from mitosis, so a lot of the portions of it are very, very similar, but there are some major differences that we need to make sure we pay attention to. All right, so here is meiosis. We call the phases the exact same thing. We just put numbers off after them because everything happens twice. So there's interphase 1, prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. So it's IP on the map, P on the map. Yeah, you just have to remember that everything happens twice. And then the other thing is when you're talking about meiosis, you have to specify. You can't just say, oh, it happens in prophase, because there's 2 of them. You have to say if something happens during a portion of meiosis, you have to say, does it happen in the first part of it or the second? So prophase 1 or prophase 2. If you only said prophase, whatever the question was, you would have the wrong answer because you cannot, the person wouldn't know if you knew the difference between prophase 1 and prophase 2. All right, so how does this work? What's going on here? So first thing that happens is that the DNA replicates, okay? So you start out, so this cell has only 2 chromosomes in it, and it replicated and made 2 X's, okay? And then the first half of meiosis, the first division is going to separate the homologous pairs. So you're going to separate out so that you pass on information either that you got from your mom or information that you got from your dad, okay? And then the second division is going to separate sister chromatids. The second division looks a lot like mitosis, okay? So first division separates homologous pairs, second division separates sister chromatids. You've got to remember that. All right, so let's go through it a little bit more. So in interphase of meiosis, first off, the DNA is replicated. Okay, that might seem strange since we're trying to go in half. We go double and then half and then half. Seems pretty inefficient, and it is. But you have to remember that meiosis evolved from mitosis. So the body tries to be as efficient with its evolution as possible. So it's just using the same machinery of mitosis. Okay, so the DNA is replicated in the S phase of interphase of meiosis, just like in mitosis. Then the first division is going to separate those homologous pairs. Okay? When the homologous pairs, the double-stranded chromosome from mom and the double-stranded chromosome from dad that both carry the same information, they're going to come together during prophase 1, and they're going to form this thing called a tetrad. And it's a tetrad because there's 4 chromatids here. 1, 2, 3, 4. Okay? So these tetrads are going to form first during prophase 1. Okay, they're going to line up in the middle of the cell during metaphase 1, and then the 2 X's are going to be separated from each other during anaphase 1, making 2 cells that each have double-stranded chromosomes. Okay, they're diploid cells because it's only 1 set. You only have a set from either mom or dad, but you have double-stranded of everything. So that's a little confusing that it is haploid, but it is double-stranded haploid. Okay. First division separates homologous pairs. Have to remember that. All right, then in the second division, those sister chromatids are going to line back up during metaphase 2, get separated during anaphase 2, and make new cells during telophase 2 and cytokinesis 2. So second division separates sister chromatids. And this looks just like mitosis, really. Okay, but you end up with, the end result is different. You end up with 4 cells instead of 2 cells, and those 4 cells have half as many chromosomes as the ones that you started with. Okay, we draw little tails on them to represent that these guys would be sperm, little sperm cells. Okay, so let's, a little overview here. All right, interphase, prophase 1, metaphase 1, anaphase 1, telophase 1. Separating homologous pairs, going from 2N to 1N. Then all the 2s, prophase 2, metaphase 2, anaphase 2, telophase 2, separating sister chromatids, so that you have single-stranded chromatids, and it's a lot like mitosis. All right, so here's some pretty pictures. Okay, prophase, metaphase, anaphase, then metaphase 2, anaphase 2, telophase 2. Notice that in most animal cells, the final cytokinesis, like the final actual separation, doesn't happen until telophase 2. You kind of get this ball of that kind of looks like the beginnings of 4 cells, and then 4 cells, and then they'll really separate out into each of their 4 individual cells. So cytokinesis pretty much happens at the very, very end. Okay. There's one other really important thing that happens during meiosis that does not happen during mitosis, and it's called crossing over. Okay, and it happens during prophase 1. If you're ever asked, when does crossing over happen, and you write down prophase, it is wrong. It happens during prophase 1. Okay, when the tetrads form, okay, the chromosome that this cell got from its mom and the chromosome that this cell got from its dad get together. These little tails and ends of the chromosomes sometimes will get so jumbled up that they switch places. Okay? So a portion, they're going to swap little pieces. A portion from this red chromosome is going to pop off and go onto the blue chromosome, and the portion from the blue is going to go over to the red. So you get something like that. Okay? So here's what happens. First there's the crossover, then the DNA breaks, and then it refuses. And it refuses usually in a new spot. Okay? Why is this so advantageous? Okay? It creates more variation, new combinations of traits that may have never existed on the planet before. And that might make one person so much better able to survive and reproduce that they lead to a whole new type of species. Very cool. All right, so let's look at mitosis versus meiosis. We start out with our parent cell that has 4 chromosomes. Okay? 2 that this individual got from its dad, 2 that this individual got from its mom. Okay? So the diploid number of this cell is 4. The first thing that happens in both situations is that the chromosomes replicate. Okay? Oops. Now, in meiosis, you're going to get the homologous pairs lining up, crossing over happening, homologous pairs separating, and then sister chromatids separating. In mitosis, you just get single file line, separate them out. Okay? You get 2 cells that are identical to what they came from, versus meiosis, you get 4 cells that are all usually different from each other and have half as much information as what they started with. So you see this original one has 2 longs and 2 shorts, and each of these has 1 long and 1 short, 1 long, 1 short, 1 long, 1 short, 1 long, 1 short. Okay? Always has to be that way. If one of these had 2 longs and one of these had 2 shorts, well, that would mean that this cell had the genes for, let's say, hair color, eye color, and skin color, but was missing the genes for nose shape and mouth shape. So it'd be great to have hair, eyes, and skin, but it would suck to not have a nose or mouth. Then the other one would be the opposite. It would only have nose and mouth, but it wouldn't have any of the other 3. Okay? So that's why the homologous pairs have to line up first, so that you make sure you pass on 1 for hair color, eye color, and skin color, and 1 for nose shape and mouth shape. And then the other side gets the same thing. Okay, so 1 division versus 2 divisions in mitosis and meiosis. Daughter cells are identical to the parent in mitosis, but they're all genetically different from the parent in meiosis, and they're usually genetically different from each other as well. 2 cells versus 4 cells, diploid to diploid versus diploid to haploid. Okay, remember, mitosis is happening throughout your entire life, all over your body for growth and repair. Meiosis is only happening in your sex organs, either your ovaries or your testes, and it's only happening to produce gametes. There's no crossing over mitosis, but there is in meiosis. All right, so mom's ovarian cells go through meiosis to make eggs. Dad's testicular cells go through meiosis to make sperm. These 2 come together and fertilize to make a 46 chromosome offspring. that goes through mitosis then to get bigger and bigger and bigger and bigger and bigger until it's a big old fetus, and then it's ready to be born. Okay? All right, so again, why is sexual reproduction so important? It increases genetic variation, and as we get and talk about evolution, genetic variation is the #1 most important thing for a species to have. Okay? So because of the fact that you never know how the chromosomes are going to line up on the metaphase plate, are they going to be all moms on one side and all dads on the other? Or are they going to go mom and dad, dad, mom, 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 dad, dad? You never know what order you're going to pass on. So what percentage of genes that you got from your mother versus what percentage of genes that you got from your dad are you going to pass on? Okay, so if you see, depending on how they line up on the metaphase plate, you can get all sorts of different combinations. Okay, then crossing over is going to mix then information. So maybe you're passing on one whole chromosome that you got from your dad, but like for this long one, but then this medium-sized one crosses over a bunch, and you pass on about 30% of information from mom and 40% information from, or 30 and 70, sorry, 30 plus 70 is 100, 30% information from mom and 70% information from dad. You never know what could happen. So that makes an infinite number of possibilities. Then there's also random fertilization. Which one of these is going to meet up with somebody else's gamete to make a baby? Okay, which sperm fertilizes which egg? All of these things lead to more and more and more variation, providing the information needed for natural selection. Okay? So offspring never have the same combination of genes as parents do. Okay, so you got this gamete from mom, this gamete from your dad to make you, but then you pass on some combination of mom and dad or some other combination of mom and dad. Okay, so because of random assortment, we get 2 to the 23rd different combinations. Amazing. Okay, then crossing over is going to create an infinite number of possibilities. So, and then random fertilization, which egg and which sperm are going to get together. So 2 parents will produce a zygote with over 70 trillion possible diploid combinations. So that's why siblings certainly look alike, but are not identical, unless they came from the same egg. Okay, it allows us to have both genetic similarity and differences within a family. So here's the Baldwin brothers, Alec Baldwin, and then his brothers, that's, oh, I don't remember, Stephen. That one's Billy. I don't remember. And then there's the Jonas brothers. And then there's Martin Sheen and his sons Charlie and Emilio. And yes, I know that's not really a Jonas brother, it's just supposed to be funny. Okay? All right, we'll talk about specific sperm and egg production when we get later to reproduction. So you can, for right now, ignore the rest of this PowerPoint. Okay? Hope that makes sense. See you guys next time.